everyone, I am Sonali. Welcome to our channel Success EduTech. Today we will see about plants, microbes and human welfare. Please watch complete video and please like, share, subscribe to our channel. Please watch the previous videos. Link is given in the description. Now we are going to learn about strategies for enhancement in food production and how the plant breeding helpful in disease resistance. So plant breeding is helpful in creating a disease resistance plant. So as we know that plants also suffering from many diseases like fungal, bacterial and viral diseases, and it can affect the yield of the crop and it will decrease the crop production in tropical region also. Now this is the disease which cause to the plants, and this is the citrus disease which cause to the citrus plant. Now in this situation the methods are helpful, that is plant breeding and the development of cultivars which will resistance to the disease enhance, and it will form a food production, more food production that we will want in a more quantity. Now suppose this is bacteria and this is the DNA extract from bacteria and the cloning method in which the fragment from these insert into the another DNA and it will transform. This is the transformation method where it multiply, and it is the culture method where we will culture the tissue culture technique, and it will insert into the plant which create the breeding method. Means from one plant we will get a variety of the crop plant. This is the cultivar method where we are sowing the seeds or anything. This is known as the cultivar method. Now here this also helps to reduce the dependence on the use of fungicides and bacteriocides. Means suppose the plant is the disease free or it is the disease resistance. Plants have the character which is the disease resistance character. So it will increase the use of fungicides and bacteriocides also. Now resistance of the host plant is ability to prevent the pathogen from causing disease and is determined by the genetic constitution of the host plant. Means resistance of the host plant means suppose the host plant have the characteristic to resistance from the diseases, so it will prevent it from the pathogen which can cause the disease, and it will help in a genetic constitution of the host plant also. Now here the before breeding is undertaken, it is very important to know about the causative organism. Means which organism, fungi, bacteria, viral, which organism can cause the disease, and the mode of transmission is very necessary. Now some of the following plant diseases are given here. Please see that fungal diseases, for example, brown rust of the wheat. This is the brown rust which can cause to the wheat plant. Now the red rust of the sugarcane, this is the red rot of the sugarcane disease, and the lead blight of the potato plant. This is the dark spot which can cause the lead blight of the potato plant. Bacterial disease, for example, black rot of the crucifer or citrus canker. Now see, this is the black rot of the crucifer and this is the citrus canker disease which can cause to the citrus plant. Viral diseases, for example, tobacco mosaic virus, turnip mosaic. See, this is the tobacco mosaic and the turnip mosaic virus. This disease also can cause to the cabbage. Now, method of the breeding. Now, breeding is helpful, but which method are very important for the disease resistance to create a disease resistance plant? Breeding is carried out by conventional breeding. Conventional breeding method is nothing but the disease resistance is that include the hybridization and the selection criteria. Here the wild relative plant and here the crop plant. Suppose we will make the hybridization method here. This will form a conventional breeding technique and we will get the plant like this characteristic. Now mutation breeding is the another method. I will explain it later. See, the steps are essentially identical to those for breeding of any other agronomic character such as high yield. To get the high yield, these agronomic character are very important in the plant. The various steps involved in conventional breeding are: first step is the screening germplasm for disease for resistant sources. Now this germplasm is nothing but the genetic material of the germ cell. It will we have to generate a genetic material of that plant which can help to create a variety of the plant of the disease free plant. Hybridization is technique. This technique is very important. Selection and evaluation of the hybrid is the latter technique. Testing is very important technique after that. Some crop varieties breed by hybridization and selection for disease resistance to fungi, bacteria and viral diseases. So the plant which have the disease resistance can resistance to be the diseases like fungi, bacteria and viral diseases. Now this is the table given here. The crop like wheat and variety is himgiri, and this plant is resistance to the disease leaf and stripe rust, hilburn. Then brassica pusa swarnim is the variety and resistance to the white rust. Cauliflower varieties pusa shubra, pusa snowball, k1 and resistance to the disease like black rot and curl blight, black rot. Cowpea variety pusa komal and bacterial blight. Chili variety pusa sadabahar, and the disease resistance plant of the chili are resistance diseases are chili mosaic virus, tobacco mosaic virus and leaf curl. Now here is the himgiri variety of wheat is resistance to leaf and stripe rust and hilburn. See, this plant is resistance to these diseases like leaf and stripe rust, hilburn. Then pusa swarnim karandrai variety of brassica is resistance to the white rust. This is the white rust. Pusa shubra and the pusa snowball k1 varieties of cauliflower are resistance to black rot and the curl blight, black rot. Now, pusa komal variety of cowpea is resistant to the bacterial blight. Now, here the pusa sadabahar variety of chili is resistant to chili mosaic virus, tobacco mosaic virus and leaf curl. Now, conventional breeding is often constrained by availability of limited number of the disease resistance genes and are present and identified in various crop varieties or wild relatives. Now, inducing mutation in plants through diverse means and then screening the plant materials for resistance sometimes lead to the desirable genes being identified. So, for the disease resistance plants, we have to identify the desirable genes which are resistant to the diseases. Plants having desirable characters can then be either multiplied directly or using the breeding to increase the crop production. Other breeding methods that are used in selection among somoclonal variants and the genetic engineering are very important technique in the plant breeding method. Mutation is the process by which genetic variation are created through change in the base sequence within genes. Means in the genes, in the DNA, it will create a change in the base sequence, that is genetic variation, and that result in the creation of new character. We will get the plant with new character which not found in the parental type. So we will get the new plant here. It is possible to induce a mutation artificially through the use of chemicals or the radiation. Means also we can naturally also we can produce and artificially by using chemical and radiation like gamma radiation to help in the producing by artificially and selecting and using the plant that have the desirable character as a source in breeding. This process is called mutation breeding. Now, in mung bean, resistance to yellow mosaic virus and powdery mildew were induced by mutation. Just for example is mung bean. See here, this is the mutation breeding. Now, several wild relatives of the different cultivated species of plants have been shown to have a certain resistance character but have very low yield. Hence, there is a need to introduce resistance gene into high yielding cultivated varieties. So, resistance to yellow mosaic virus in Bendi was transferred from a wild species, resulted in production of new variety called Parvani Kranti. So, from wild variety, gene transferred into these Bendi plants, and we will get a resistance plant, that is the Parvani Kranti. This is the Bhendi and Parvani Kranti. See the difference between these two pictures. Now, all the above example involve sources of resistant genes that are the same crop species or even related wild species. Transfer of resistant gene is achieved by sexual hybridization method between target and the source plant followed by the selection. So, students, all these are the technique useful in the plant breeding. So please subscribe to our channel, and thank you very much for watching the videos.